Hello student, this is my third lecture on general introduction to drugs. So these are the points that we had covered in the last lecture, that is classification of drug. You know that the drug has been classified into four broad types depending upon its therapeutic action. So the first class is functional or pharmacodynamic agent, second one chemotherapeutic agent, third one vitamins and hormones, last one is hormones. Then again in the last lecture we had learned nomenclature of drug. So the drug having three or more names like your chemical name or systematic name, generic, official or a common name or a non-proprietary name, and then third one is proprietary, trivial or a brand name or a trade name. So you know that this chemical name is given by on the basis of IUPAC nomenclature. So it describes the chemical structure of the drug. Then second one is your generic, official or a common name. So it is given by WHO. So this WHO has a right to select, approve and disseminate the generic name of the drug. So this generic name should be simple, concise, meaningful and easy to remember, but often it is not. Then again in the last lecture we had learned the definition of some medicinal terms that is pharmacone, pharmacology, pharmacophore, then prodrug, then half-life efficiency, LD50, that is median lethal dose, then ED50, median effective dose, GI50, growth inhibition of 50% of cell, and therapeutic index. So these are the points that we had covered in the last lecture. So dear students, today we are going to learn some brief idea of the following terms. So the first term is receptor. So this receptor, it is a portion of molecule or a structure with which the therapeutically active compounds interact, producing series of events leading to a observable response. So this is receptor, means it is a portion of molecule or a structure and that can interact with the therapeutically active compound leading to a observable response, that is called as receptor. So some structurally specific drugs, after interacting with specific cellular component known as the receptor, so it form a complex with that receptor, and chemically receptor is a structural component and biologically receptor is a micro anatomical term. So I'm saying that chemically receptor is a chemical structural component and biologically receptor is a micro anatomical term. So receptor is nothing but it is a portion of molecule or a structure with which the therapeutically active compound, that is drug, active compound drug interact with it and producing a observable response, that is called as receptor. Then next point is agonist molecule. So it is a chemical or a drug that binds to a receptor and activate the receptor to produce a biological response. So agonist molecule is nothing but it is a chemical or a drug that bind to a receptor, that bind to this receptor and it activate that receptor to produce a biological response. So these are the example of some drug that having that act as an agonist molecule. So this agonist molecule activate the drug and to produce the biological response. Then second term is antagonist molecule. It is a chemical or a drug that bind to a receptor and produce a biological response. So the agonist molecule, these are the chemical or drug that binds to a receptor and blocks the biological response. So these are the examples of antagonist molecule. So it bind to a receptor and it blocks the response of that receptor. So we can convert by changing or by introducing or by removing some functional group to agonist to antagonist molecule. The next point is drug receptor interaction. So the drug action results from physiochemical properties in case of structurally non-specific drug, but in case of structurally specific drug, their action result from the chemical structure. Drug action is due to binding of drug to the receptor site. The drug binding is due to ionic and non-ionic forces. Non-ionic binding is due to hydrogen bonding, hydrophobic bonding, and van der Waals forces. The interaction involves the formation of drug receptor complex and the resultant response or a effect. So here in this where D is drug, R is a receptor, and this Dr is a drug receptor complex, where E is a biological response, and K1, K2, K3, these are the rate constant for absorption, deabsorption from the receptor side, and the production of receptor effect respectively. The most important factor determining the drug action is the rate at which drug combine with receptor. So a chain of events leads to a observable biological response by formation of drug receptor complex. Drug receptor action at the receptor level is proportional to the number of receptors occupied in a certain site of the organism. Drug structural feature contribute to its affinity for the receptor and efficiency of the response, that is intrinsic activity. Agonist and antagonist molecule passes structural feature which promotes drug receptor action. However, only agonist molecule passes stimulant action, that is intrinsic activity. So only agonist molecule passes some functional group which passes some stimulant action which activate the drug. Then one important point is antagonist molecule also bind strongly to the receptor, but they inhibit the activity of a receptor. The agonist and antagonist molecules can be interconverted by removal or addition of some certain chemical groups like alkyl group. Antagonists have strong binding properties due to high rate of association but low rate of dissociation. So this antagonist molecule having high rate of association, and it deactivate because it having low rate of dissociation. When the receptor site is blocked by the antagonist molecule, it prevents the productive action by the other molecule, and this is the principle of occupancy theory. So what happen is that here agonist having high antagonist molecule having high binding capacity, so it can bind with that receptor, but it having low rate of detachment or low rate of dissociation. So that's why it deactivate that receptor. The next point is drug potency. So the drug potency can be defined as the maximum biological action which can be caused by the minimum amount of the drug administered. So by using minimum amount of drug which can give you the minimum amount of high biological response, that is called as drug potency. The drug potency can be defined as the maximum biological action which can be caused by minimum amount of drug administered. A highly potent drug can cause high biological effect even with a minimum dosage. Thus the potency is inversely proportional to the dose. The potency also depend on the body weight. It is often expressed as body weight per unit weight of the drug to cause a particular effect. Thus a drug which is highly potent to children may not have a good effect on adults. Potency can be used to compare the relative activity of a series of drugs having same physiological activity. So by using this potency you can relate the activity of a drug having same physiological activity. For example, both codeine and morphine. So we have taken the example to understand what is drug potency. For example, both codeine and morphine are used as a pain reliever. However, even large dose of codeine cannot effectively relieve the pain, severe pain, which can be relieved by a smaller dose of morphine. So thus you can say that the morphine has a higher potency because it is effective in a smaller dose. So thus the morphine has a higher potency because it is effective in a smaller dose. If the concentration of the drug increases in the vicinity of the receptor, then the biological response will be increases. The biological response resulting from various dosage of the drug may be represented as a DRC curve. So this is your DRC curve. So in this figure, the dose response curve for this three drug 1, drug 2 and drug 3 indicates the dependence of response on the concentration of drug in the area of the receptor. Initially, the higher concentration of the drug rapidly increases the response. But at higher concentration of the drug, the extent of response is no longer increases, because at this point most of the receptor required to produce a response have a combined with a drug. So this DRC curve indicates that drug 2 provide full response only at higher concentration than required for drug 1. Hence drug 2 is less potent than drug 1. Similarly, this drug 3 is less potent than drug 2 because it shows higher response only at higher concentration. So this decrease in the potency of this drug 2 and drug 3 is due to it may be due to less efficient distribution to the receptor. So that drug may have less efficiently distributed near to that receptor. The next point is less affinity for the receptor. So the drug may have less affinity for that receptor. So this also be the reason. So that's why that this drug don't have high potency. Then the last one is greater rate of metabolism or excretion. So this the drug may be excreted before it absorb in your body. So these are the three different reasons why the drug having less potency. So next point is bioavailability. So the bioavailability is defined as the percentage of biologically active drug which is absorbed from a given dosage form and reaches the systemic circulation in the body following the administration of drug. So it is the concentration of drug in your body, or it is the percentage of biologically active drug which absorbed from a given dose and shows the biological response, that is called as bioavailability. So it depends. So this bioavailability depend on route by which it is administered. When the drug is given by intravascular route, the bioavailability is 100%. However, it will be much less in case of oral administration. When it is given by oral route, the drug may not be fully absorbed or may not undergo change, or it may undergo change in a liver or intestine, or it may be excreted before it absorbed. So these are the reason why the drug having less bioavailability. So the reason is it may undergo metabolic changes in the liver or intestine, or it may be excreted before it absorbed. So the bioavailability can be found by measuring the concentration of drug in a blood or a urine sample at a regular interval and plotting the concentration time curve. Thus, two similar pharmaceutical may not have a same bioavailability and may not provide same therapeutic benefit. So there are certain factors which affect the bioavailability that we are going to see. That is, first one is the properties of the drug like particle size, solubility, hydrated form, and the metal salt use, and the chiral characteristic may affect the bioavailability. For example, the water-soluble drugs have a higher availability as they are easily absorbed. In case of chiral molecule, if racemic mixture is used, then the bioavailability is less than 50% because only one of the isomer is biologically active. So this is the reason if you are using racemic mixture. So in racemic mixture you are having two isomer, two enantiomer. So one enantiomer will be active and one is inactive. So that's why you are having only 50% bioavailability because only one isomer is biologically active. And another one is if what your drug is having high water solubility, so it may be easily absorbed. Then the next point is the pharmaceutical preparation of some additives and excipient are added. So while preparing the drug, we are adding some additives and excipient. For example, dyes, preservative, base, antioxidant, and other like sodium sulfate. So this may interact with your drug molecule. The sodium salt of carboxymethyl cellulose (CMC), which is used as a diluent, is found to be complex with some antihistamine and tranquilizers. So all this affect the bioavailability. So the best example is the sodium salt of CMC, which is used as a diluent, is found to be a complex with a some antihistamine and tranquilizer drug. So that's why it affects the bioavailability because your drug is forming complex with this CMC. So that's why the bioavailability is less. So all these factors affect the bioavailability. The next point is the granules of the drug are mixed with binding and fragmenting. The granules of the drugs are mixed with binding and fragmenting agents and compressed into a tablet and then coated with a suitable material. So the bioavailability depends on how these processes are carried out. For example, microfine aspro break into fine particles due to disintegrating agent added. So this increases the absorption of the drug, hence bioavailability increases. Then next point is drug toxicity. Drug toxicity. Drug administration is always followed by expected reaction considered to be toxic reaction. This may range from skin rash to more serious complication like blood discrepancies. It means that the abnormality in blood picture, damage may occur. A drug shows short-term acute or a long-term chronic toxicity. So so many drugs having this toxicity. So this drug may have range of toxicity. So it may start from skin rash to more severe complication like blood discrepancies, that is abnormality in blood picture. Then next point is drug addiction. So after discontinuation of administration of drug, tolerance acquired by the individual disappear at a rate varying for each individual or for each drug. So what is drug addiction? I'm going to tell you in a simple language. In some cases, what happen after the drug withdrawal or a psychic craving for the drug is observed, which leads to a physical disturbances. So this syndrome is called as addiction. Certain drugs, for example morphine related compound like cocaine and certain barbiturates, are known to be cause addiction. So this cocaine and barbiturates are a addiction causing agent. It causes the addiction. So this is all about bioavailability, then drug toxicity and drug addiction. Drug addiction is nothing but in some cases after the drug withdrawal a psychic craving for the drug is observed, which leads to a physical disturbance. This syndrome is called as addiction. Certain drugs, for example morphine related compounds, cocaine and certain barbiturates, are known to be cause are known to be cause addiction. Then the next point is, which drug is called as spurious drug? A drug shall be deemed to be spurious drug if it is imported under the name which belongs to another drug. That drug is called as spurious drug. If the drug imported under the name which belongs to another drug, that drug is also considered as a spurious drug. Then second point is, if imitation of or a substitute for another drug for resemble another drug in a manner like it disuse or it bear it upon or it label or a container the name of another drug, unless it is plainly or conspicuously marketed so as to reveal its true character or lack of identity with such other drug, that drug is also spurious drug. If it contains substitute or the same like another drug, or if it contain any label which resemble to another drug, or that drug is called as spurious drug. So these are the criteria to find out whether the drug is spurious drug or not. The next point is, if the label or container bears the name of an individual or a company purporting to be the manufacturer of the drug, which individual or company is fictitious or does not exist. So spurious drug means if the label or a container bears the name of an individual or a company purporting to be the manufacturer of the drug, which individual or a company is fictitious or does not exist. If you are having label, that label or individual label, if it bear label or a container bears a name of an individual or a company purporting to be the manufacturer of the drug, which individual or a company is fictitious or does not exist, that drug is also called as spurious drug. The next point is, if it has been substituted wholly or in part of any other drug, that drug is also called as spurious drug. If any part of the drug substituted by another drug, so that drug may be a spurious drug. Then the next point is, if it purports to be the product of manufacturer whom it not truly a product, so that drug is also called as spurious drug. If it purports to be the product of manufacturer of whom it not truly a product. Then next point is misbranded drug. Which drug is called as misbranded drug? So there are certain criteria to find out whether that drug is misbranded or not. So a drug shall be deemed to be misbranded drug if it is so colored, ordered, polished that damage is cancelled, or if it is made to appear better of or a greater therapeutic value than it really is, that drug is also called as misbranded drug. If it is so colored, ordered or polished, or that damage is cancelled, or if it is made to appear better or greater therapeutic value than really it is, that drug is called as misbranded drug. And then next criteria to find out whether that drug is misbranded or another drug. So if it is not labeled in the prescribed manner, if your drug is not labeled in the prescribed manner, that drug is also misbranded drug. The next point is, if it's label or it's not a label, it's a label, if it's a label, it's not a label, if it's a label or a container or anything accompanying the drug bears any statement, design or device which make any false or misleading claim for the drug, that drug is also called as misbranded drug. If it label or a container or anything accompanying the drugs bear any statement, design or device which make any false or misleading claim for the drug, that drug is also called as misbranded drug. Then next point is adulterated drug. Which drug shall be deemed to be adulterated drug? if it consists in a whole or in any part of any filthy, putrid or decomposed substance. If it consists in a whole or in part of any filthy, putrid or decomposed substance, that drug may be adulterated drug. Then if it has been prepared, packed or stored under unsanitary, if it is packed under unsanitary condition, whereby it may have been contaminated with filth, or whereby it may have been rendered injurious to health, that drug is also called as adulterated drug. If it has been prepared, packed or stored under unsanitary conditions, whereby it may have been contaminated with a filth, or whereby it may have been rendered injurious to health, that drug is also called as adulterated drug, or that drug may be adulterated drug. The next point is, if its container is composed in a whole or in part of any poisonous or a harmful substance which may render to contain injurious to health, so that drug may be adulterated drug. If it contains any harmful or toxic substance which may render it injurious to health, that drug is an adulterated drug. Then if it bears or it contains color other than one which is really approved, if your drug having another color which is really other than the one which is really approved, that drug may be a adulterated drug. Then if the drug has been mixed with any other substances so as to reduce its quality or strength, that drug may be adulterated drug. So these are the some points to find out whether that drug is adulterated or not. The next point is pharmacopoeia. What is pharmacopoeia? So this pharmacopoeia is made up of two different words. Pharmacon means drug and poem means to make. So it is a book of direction and requirement for the preparation of medicine. It is generally published by an authority. Thus, pharmacopoeia is the legislation of the country which sets the standard and obligatory quality indices for the drug, raw material used for the preparation, and various pharmaceutical preparation. So these regulations are presented separately in general and for specific article. Only the pharmaceutical which are commonly and currently in use are included in the pharmacopoeia. The substances which are found to be undesirable from the past and experience and which are currently not in use are excluded. Only the chemical which are used for application and internal consumption by human being from the part these are the part of pharmacopoeia. In pharmacopoeia, only minimum standard for chemical are prescribed. However, the manufacturer may supply this with more stringent standard. Thus, the medication has to comply strictly this with any one of the pharmacopoeias. If not, it may be considered standard drug and unstandard drug or substandard drug, and usually not prescribed by standard medical practitioner. Only those mentioned in the pharmacopoeia are considered official as per the legislation of the country. Then the next, so these are the pharmacopoeia depending upon the country. So first one is British Pharmacopoeia. So British Pharmacopoeia was the pioneer in this field, having published their first edition in 1883. Since then till today, the British Pharmacopoeia is rated as the most standard pharmacopoeia. In Britain, only pharmaceutical manufactured as per the BP standard can be marketed. In India, before independence, BP was used as the official book of standard before independence. In Britain, the manufacture and marketing of the drug and pharmaceutical is subjected to stringent legislation. These medications are not as per the standard are withdrawn. The medication which are not standard as per the BP are withdrawn. Then next one is the United States Pharmacopoeia, that is USP. The first USP got released on 15 December 1820. It is exhaustive and it covers all pharmaceutical chemicals, raw materials, drugs, formulations, chemicals, then your vitamins, hormones and blood products. In United States, medicine which are not as per USP specification are not allowed to be marketed, nor they are prescribed. United States generally does not import medicine and food products unless they are made as per USP norms. The next one is our Indian Pharmacopoeia. So the first edition of the Indian Pharmacopoeia, that is IP, got published in 1955. It was having a large number of crude drugs and their preparation. The general notices and appendices includes in the Indian Pharmacopoeia in 1985 and as amended in the addendum apply both to the matter contained in the Indian Pharmacopoeia in 1985 and to the matter contained in this addendum. The third edition of Indian Pharmacopoeia was published in 1985, which was amended by addendum first in 1989. Each later edition supersedes the previous one. There are also supplements, means addendum 1 each in, that is in 1960, 1975, 1990, 1992, in order to keep the pharmacopoeia accurate, authoritative and up to date. The ministry has established the Central Indian Pharmacopoeia Laboratory at Ghaziabad and the Indian Pharmacopoeia Committee. So this is all about the Pharmacopoeia, that is British, United States Pharmacopoeia and Indian Pharmacopoeia. Thank you.